So now that we have left behind our evolution chapter, we sort of transitioned from evolution to this section through the exam with that chapter on phylogenetics. Right? It was a little bit of information where we kind of looked at the tree of life and we thought about evolutionary relationships. And what we're going to do now is kind of go up that tree of life. Okay? And so we'll spend this entire middle exam on what I call the diversity of life. So we're going to go through all of the different diversity. We'll probably we're going to move fungus. I had fungus before animals. I'm going to move it to after animals. We'll go out in the woods and find some good fungus anyway. And then probably fungus and maybe plants both will actually be on the last exam, but we'll get through acellular, single cellular, and heterotrophic eukaryotic life for this exam. Okay, which is about 4 chapters. I'm going to treat chapters 21, 22, and 23 as one for the purposes of homework and quizzes, because we are doing a fraction of each of those chapters. And with everything that we're going to do for this, I mean, this is true of everything, but in many cases, like each of these chapters have their own class, right? Like prokaryotes, that's microbiology. When we talk about all of the invertebrate diversity, there are like entire invertebrate zoology classes that are upper level classes. So we're going to have to, kind of crunch a ton of stuff. And I just want to give you some idea of the breadth of diversity. So we're going to stick to that very skimming the surface layer. So there's a lot of information in your book I'm not going to cover in these chapters, and it's interesting, and you can feel free to read it. But like in terms of what I'm going to test you on, remember, I'm only going to test you on stuff from the slides. Okay, that's what I'm going to ask you about. So chapter 21, we're going to cover a really small amount of the information. And so we're going to talk about acellular life, right? This is this weird place where we're kind of between stuff that isn't alive and stuff that is alive. And we're not really 100% in agreement about what we want to, how we want to characterize these things. Generally speaking, we don't think of them as living organisms because they are incapable of living independently, yet they evolve. And so, where do you put that? Okay, so I'm going to call it acellular life, but we're going to use air quotes around life, because if you go back and think from Bio 1, your characteristics of life, these organisms just don't have all of those characteristics. So they're in this weird place in between. We're mostly talking about viruses, and then we're going to also talk a little bit about these other things, prions and viroids, which are even weirder in some ways than viruses. So the whole chapter of stuff we don't really know how to categorize, and it doesn't have a place on our tree of life. Like, it's not in there. It's not a prokaryote. It's not a eukaryote. It's just not even there. So there are multiple hypotheses about where viruses originate from. It talks about them in a really superficial way. I don't think we really know for sure where they originate from. There are, I think, like 3 competing hypotheses, all of which are potentially possible, but we don't really know. And so they don't have a clear linear evolution the way we think about when we talk about our tree of life, yet they are, I mean, like the flu shot changes every year, right? And they're trying to predict what they think should be in the flu shot each year. And that's because the viruses that make up influenza are constantly changing, and they are adapting. And so they're experiencing adaptive evolution to the surrounding environment, but it is not necessarily, like, it's responsive as opposed to some sort of directional thing, where most of our tree of life is like kind of starts off in a direction and goes there. I feel like viruses are much more fluid in how they shift, and they're very responsive to what's happening in their hosts, right? And so they're host driven as opposed to independently driven. I did not answer your question at all. So I recognize that. Like, never answer your questions. Are you dissatisfied? Do you want a refund? Okay, so I want to start by giving you guys a little bit of a frame of reference for thinking about these particles, these like acellular things. This is the very edge of a eukaryotic cell, right? Nucleus, endoplasmic reticulum, Golgi, mitochondria, right? That gives you a rough idea of eukaryotic cell. Then here we have your sort of typical prokaryotic cell, E. coli down here. And so this is sort of typical prokaryotic cell size, roughly the same size, roughly equal to a mitochondria. The prokaryotes that we looked at last week in our single-celled life lab were big prokaryotes. This is a cyanobacterium. And so the photosynthetic prokaryotes are huge in comparison to the non-photosynthetic counterparts. They're much larger. Here is another standard bacteria, Staphylococcus. It has a different shape than E. coli. It's not as long. It's round. Most bacteria we're going to talk about sort of fall into one of three shape categories, but they're still more size to our E. coli than they are to like the cyanobacteria. The stuff we're going to talk about in this chapter is over here. Okay, so viruses and prions and viroids are just tiny. They're pieces of molecules. Okay, they're much, much smaller than a cellular complex single-celled or multicellular organism. Okay, I'll come back to this slide a couple of times, just a frame of reference. I'm not going to ask you how big any of these things are. I can have you report micrometers to me. Okay. So the best way I could come up with to define viruses is to call them an acellular parasitic entity. Okay. I'm not sure that that's the only way we could define them, but I think that's a pretty good way. Okay. They are not made up of cells, and they are obligate parasites. They cannot function and live independently of a host. Okay. They can survive independently of a host. They can lay dormant for long periods of time. In fact, a lot of the climate change models predict some potentially really scary stuff happening as viruses that we haven't experienced in hundreds of thousands of years or more start to emerge as the ice melts. They may still be viable. In fact, there's good evidence that they will be many of them will be viable. And as soon as they find a suitable host, again, they could take off. So they're sort of in this morass between living and non-living entities. They are made up of genetic material surrounded by a protein coat. Now, when we talk about life, we always say that DNA is the genetic material of life, right? Viruses can either have DNA or RNA as their genetic material. So they're not beholden just to DNA as their background genetic material. And they use host cells to carry out their metabolic functions. So they can't reproduce without a host. They can't make new materials. They rely on the host to do that work for them. Okay, so they infect a host and then take over the host's cellular machinery and have that machinery do the bidding of the virus, basically. So these are just a couple of different viruses. They come in different shapes and sizes. We're going to look at the bacteriophage a little bit, tobacco mosaic virus, so plant virus, and then the influenza virus, which many of us are becoming quite intimate with this fall or this spring, right? But just 3 different shapes. Okay. We can also talk about them sort of being separate from their host, and we usually use the term viron when we have like a single viral particle that is outside of a host. And so in this particular image, I love this image. I think I use it every chance that I have. This is the boundary of a bacteria cell. And all these guys that look like little moon landers are bacteriophages. So they're viruses that infect bacteria. And they have this awesome, creepy structure that's kind of like a syringe, and they're able to actually inject their genetic material into the bacteria's body. And so that's what's happening here. Like this green that you see going down in, that's the genetic material being injected from the protein coat that's on the outside into the bacteria's body. And then that genetic information will actually start controlling the operation of the bacteria. Okay. All right. So they're non-cellular. Viruses have super simple morphology. They're simply a nucleic acid genetic material. It's like the place to congregate and chat right outside our classroom. So they have this nucleic acid core, either DNA or RNA, right? And then they have an outer protein coat. And if you like just Google viruses, right? And look at all, like do an images search and look at all the different shapes. They really have a whole bunch of different shapes. And that comes from the protein coat. And the protein coat usually has a role in how the virus gets into the host cell, right? Here's our bacteriophage that I just showed you. That looks like so here is actually where the DNA is housed in this unit. And then this whole part here is all protein, and it's designed to be able to attach to the bacteria, right? And then we can inject the DNA down into the bacteria. Where some of these other things have these like outer matrices that they actually use to kind of fuse with the wall of the or the plasma membrane of the host cell, and sort of stick to it, and then they can actually merge and spill their contents in. It totally depends on how the virus functions. We're not going to go into too much detail. You can take a virology class if you want to get in the weeds with viruses.
Baltimore?
Hmm?
Is that David Baltimore?
I don't know.
Dr. David Baltimore?
I don't know. Can oh, I was gonna see if she would close the door for me. I actually don't know which Baltimore. Oh yeah, he's a Nobel Prize laureate. Yeah. I'm not a person that I know their history. But their name is on the classification scheme used for viruses. So we don't have the tree of life for these guys, right? They don't fit into our phylogenetic hypothesis. And sort of like prokaryotes, we have had some sort of way of defining and classifying them, putting them into a category. What we're starting to switch over to with prokaryotes is a more genetic-based category. And in some ways this is because it actually has to do with whether they're a single-stranded or double-stranded DNA virus or RNA virus, but it is still based on morphology as much as anything, right? What is the content of the genetic information? So there's categories, there's DNA viruses. It's kind of hard to see. There are not very many pretty diagrams of the Baltimore classification. So these are DNA viruses. These guys over here are RNA viruses. And then these are retro transcribing viruses. And there's multiple classes of each. Okay, so in our DNA viruses, we have some whose genetic material is double-stranded and some whose genetic material is single-stranded. In RNA viruses, there's actually some double-stranded RNA genetic material, single-stranded RNA that works as the messenger RNA directly, and then single-stranded RNA that transcribes complementary RNA strand as messenger RNA. Okay, so there's like different ways that that system can work. And then there's single-stranded RNA that does reverse transcription. and a double-stranded RNA that does a reverse transcription. So retrovirus has to do with reverse transcription. So you essentially go from an RNA molecule to a DNA molecule. And then that DNA is compatible with the host genetic material and can get inserted directly into the host genome. So those are sort of the categories. There are a lot of examples in each of those categories, but that's just giving you, we basically categorize them based on what is their genetic material, and then how does that genetic material work for transcription. Because really, these guys are transcription machines. That's what they do. They make copies of their genome and use it to direct the function of a host cell. Does that make sense? Okay. So they either do that by directly just using their genetic material or by transcribing a message that the host cell can use. Okay. Ultimately, we make some proteins. Those proteins are going to build the houses for more genetic material, and we assemble those viruses and send them out into the world from the host cell. I couldn't remember what I added here. So I'm going to talk about bacteriophages first, and then we'll talk about just generically some other viruses. Bacteriophages can have two different life cycles, and so if you go on and take more education on viruses, you'll talk about this quite a bit more if you take microbiology. Bacteriophages can either enter what's called the lytic cycle or the lysogenic cycle. And so we'll look at the lytic cycle first. And so here is another great image of bacteriophages on a bacteria. I don't know why I think they're so beautiful. Just are. So here we have our bacteria cell, right? Remember, bacteria has one giant circular chromosome. Doesn't have linear chromosomes like we do. In addition to their one giant genomic chromosome, they may have small subsidiary plasmids, right? But the functional genes that are necessary for the bacteria to survive and reproduce are housed in that single chromosome, okay? So our little bacteriophage lands on the surface of the bacteria's body and injects its genetic information into the bacteria. The protein coat goes away. It's not actually going to enter the bacteria. It was just the carrier to get the information there, and it's essentially done now. Okay, its purpose has served. The genetic material from the bacteriophage enters into the cell, and in the lytic cycle, the first thing that it does is it destroys the bacteria's own genome. So it essentially carries the information to disassemble that circular chromosome. So we chop it up, make it no longer functional. And now bacteria is going to just listen to the genetic material from the virus. And so it's going to make more copies of the virus's genome. And then it's going to carry out the instructions to assemble the protein coat for the virus genome. So it's going to make the little head capsules, it's going to make the little moon lander legs, and it's going to assemble those proteins. And once all of the new bacteriophages have been created, the host cell bursts, its life is over, and the virus is released out into the solution. Okay. Pretty cold, right? Viruses don't mess around. So the bacteria has been dead essentially since its genome was disassembled at this point. It's just a zombie host for the virus. And then when the viruses are ready to take off, they burst the cell and release. The alternative to the lytic cycle is to begin with what we call the lysogenic cycle. But we start off with our bacteria cell, and the cell receives genetic information from the bacteriophage. And then the bacteria cell takes that information and incorporates it into its own chromosome. And so it actually picks up that genetic information and joins it with its chromosomal DNA. And this is not an uncommon thing to happen in bacteria. We talked about horizontal gene transfer. We've talked in Bio 1 about plasmids, those accessory genes. Bacteria play a little fast and loose with picking up genetic information. And so they incorporate that into their genome, and it's called a prophage at that point. So it usually has to do with signals that are coming from the bacteriophage's genome itself in response to the environment. So if the environment is an environment that's going to be a good one for the virus to go out and populate and spread now, it'll just enter the lytic cycle. If things are looking a little slim on the bacteria or on the virus success front, they'll go for the prophage. And I actually don't know anything more finer detail than that, but it comes with how the genome enters the bacteria. It's directing what happens to it. And it's usually a assessment of like what the environment is like. So if it wants to lay low in the host, it enters this lysogenic cycle. And now every single time that bacteria reproduces, it's copying the genome. So instead of doing a bunch of copies at once and killing the host, it's just slowly spreading. Do you have a question? Yep, so getting there. Every time the bacteria reproduces, it's making a copy of its own genome and the virus's genome, right? Think about something like an E. coli at body temperature can reproduce every 20 minutes, right? So it's not like the virus isn't getting its genome copied at a pretty fast rate if it's just letting the bacteria copy. And then when conditions look good again, that prophage separates from the bacteria's genome, and the bacteria actually enters the lytic cycle. And so at that point, it functions just as if it had entered the lytic cycle straight away.
So if they're like between life and non-life, are they considered species? Like, are there other species that are different kinds?
So they're usually just referred to as strains as opposed to species. And there's classes of viruses. So like bacteriophage is like a whole class of a lot of different viruses that infect bacteria. So the bacteriophage means virus that infects bacteria, essentially. All of the retroviruses are the viruses that can take an RNA genome and reverse transcribe a DNA molecule from it. And so like they're categorized very broadly. And then we have the different strains. So like when you hear about like the flu vaccine this year, because it's sort of in the news and on our minds, like you hear about H1N1 or HN31 or all of the different names that they have, those are strains. And so those are a unique molecule type that is particularly effective this year. Whatever. So that's a great question. Did everybody hear his question? So he's asking, like, could a bacteriophage change and infect something else, right? And I can't think of a good example off the top of my head of like something changing its shape, but certainly there's some host jumping that happens, right? There are certain viruses that probably started with other primates as their hosts and jumped to humans, or started with other mammals as their hosts and jumped to humans, or started with domestic or with wild birds as a host and jumped to domestic birds or something. And so I don't know how far that goes, like if you would completely jump from a prokaryote to a eukaryote. But I also don't know that we would know, right? Like, what is the evolutionary history of a virus? Basically, I'm giving you a non-answer again. That's like, I feel like that's my shtick with all your questions. Like, I'm just going to talk around it. Owen.
Back to the flu vaccine, I know it usually comes with like
I don't know. I don't know how we decide what is the right amount, because they're not live viruses. Live viruses are a little bit riskier in terms of what the impact is on the immune system. But it probably has to do with effectiveness, like how much you can like reasonably respond to effectively to have enough immunity build up to actually mount a defense when you meet that virus. Be my gut reaction. Is there another question over here? We are so dancing around the limits of my knowledge with virology. Sorry. Huh? Yeah, these all be great research papers. Educate me about viruses if you so feel inclined. Okay. I have never taken a virology class, so caveat there. Okay. So in the case of the bacteriophage, I give that as an example because I think they're beautiful. And also because they pretty blatantly destroy the cell, right? Not every virus immediately destroys the cell. And even the bacteriophages can kind of lay low for a while. But the influenza virus actually exits the cell via budding. So the cell can continue to be a host and build more viruses or get infected again. They don't immediately sort of destroy it in order for the virus to exit. So here's our little influenza virus, and the influenza virus attaches to the target cell, typically an epithelial cell, and the cell engulfs the virus and brings it in via endocytosis. Right? It just brings it into the cell. And then the virus releases its content. And so the content actually goes out into the cytoplasm of that epithelial. And the viral RNA can get into the nucleus, and it's replicated by viral RNA polymerase. Okay. Then the viral RNA carries the instructions to make the viral proteins. So we start translating those instructions and making the viral proteins, and then assemble virus particles, which can bud off at the surface and go out and infect neighboring cells. Right? So that cell's machinery to make its copies, and then it can go out and infect the neighbors and kind of spread from there. The cell can just keep making new viruses because its membrane is not damaged by the budding process. That's natural entry and exit for a plasma membrane. So it's able to just keep serving as a host. So there's an advantage to the virus there as well, right? If you kill your host, you better have made enough copies before that death occurred to meet your goals, right? If you chronically kill your host, you run out of hosts. Right? So you have to have something like a bacteria, which is abundant enough that you can kill your host cells. Okay. Oh, that's it for viruses. See, I said we weren't going to spend much time on these. It was way more time than I was expecting to spend. Any questions that I might be able to answer about viruses? Yeah. Smart here. I'm willing to be vulnerable with you guys about this. Okay. The next acellular things we want to talk about, just really briefly so that you know that they exist, are prions. You've heard a few of you. Okay. How many of you have heard of mad cow disease before? Everybody's heard of mad cow, right? Yep. Prions cause mad cow disease, along with a whole suite of related diseases, depending on the organism. Prions are just proteins. They aren't even genetic material. They're just little infectious proteinaceous particles. So they don't have any nucleic acids. They don't have genetic machinery. They don't transcribe and translate. They're just little infectious proteins that are in some cases darn near immortal. Like they can survive in the soil for decades and still be picked up later, like by generations later, of organisms, and some of them are still functional. So they're just these sort of highly infectious, pretty stable protein particles? It's a good question. I feel like I wrote this down. Do I have a note? Nope. I think they actually, I'm drawing a blank. Let me look it up and get back to you because I don't know. I was ready to give this lecture last week and it's like since left my brain. I didn't put any notes in. It's the pond water. It's definitely the pond water. Because I had that question as soon as I was thinking about them again, and I know I looked it up. Now I can't bring it to my head. Yeah, sorry. The buildup of what? What is CTE? I don't know. Yeah, well, in Alzheimer's is buildup of a plaque protein, but it doesn't have to be foreign as in from another organism. It's usually from self-organism, but it's not usually from yourself. So, oh, that's that. So I listed off the different diseases that we're aware of that are prion caused. They're all in the same effect. They have the same effect, and the proteins are found in neurological tissue. So if you hear of chronic wasting, if anybody is in the Intermountain West or out on the West Coast, it's something that you think about a lot, especially if you harvest wild animals, because the elk and the deer all are potential carriers of chronic wasting disease. It's in the soil and it's prevalent. So if you're a hunter and you harvest an elk, you would want to very carefully remove all of the neurological tissue in the spinal cord and the brain and not contaminate any of the meat you're going to consume because the infectious particles are found in that part of the body. Bovine spongiform encephalopathy is mad cow disease. Same thing, it's brain and nervous cord tissue. And so if a human consumes that, then they can actually get that same disease. And in humans, it's called Jacob disease. Jacob disease, I think is actually the correct pronunciation. Kuru is a similar disease that was actually spread by cannibalism between humans. And so there's a particular tribe that it was fairly prevalent in that practiced cannibalism as part of their society. And it was really prolific in their populations, and they actually stopped using cannibalism as part of their ritual culture in order to reduce the instance of disease. The picture down here sort of shows you a human brain slice with Creutzfeldt-Jakob and a normal brain. And so just like you were talking about CTE or Alzheimer's, if you look at Huntington's disease, the impact on what the brain looks like is really similar. And so these proteins build up and they cause plaques and they cause gaps and normal tissue loss, and you end up with holes. And so you can sort of see you have an increase of existing holes, but I love this picture because it also shows you what happens to the folds in the brain. Like you're really just separating out all of that tissue and losing really like remarkable quantities of brain volume are lost. No, it's a different, it's, I shouldn't like throw that all in there together. No, those other diseases are not prion caused, but they also involve, like they have a similar appearance to the brain. Yeah. Awesome. Yeah. Right. Yep, and they don't use. Okay. Making normal prions or making normal proteins. Thank you. Technology in the classroom. I need my own little Let me quickly Google that. Okay. Related and similar to viruses, but not the same, are viroids. And viroids are not DNA or RNA or single-stranded or double-stranded like viruses are. They're always short, circular, single strands, and they're always RNA. And thus far, in our assessment of the viral world, viroids are only occur in plants. We have not found any that occur outside of plants. And they enter the host cell nucleus and then directly synthesize new copies of themselves using the host cell machinery. And they result in, like, this is a potato viroid, which is one of the places that it's most commonly known in is in potatoes. And it causes the potatoes to have this sort of skinny finger-like shape instead of a normal potato shape. They're not storing enough resources, and they don't thrive. And so these viroids are mostly understood because they have a pretty big agricultural impact. Yeah, there's lots of different, there's lots of different types of potatoes that we've used artificial selection to produce what we want to produce. These are actually misshapen because their tissue isn't functioning properly, as opposed to, those are all also just artificial selection. Going back to potato roots, potatoes came from South America, and South American potatoes are pretty interesting. There's lots of diversity. Some of them actually still carry more blight resistance than current Western potatoes. If it's not if it doesn't infect humans, that it would just be digested. And most things, like if you consume them, they're not entering your body in the same way they would enter if they were a pathogen. And so like your pH 2 stomach just chops it up, usually. So there's like lots of things that could be a parasite if they entered through the skin and got into the bloodstream, but are just lunch if they go through the digestive tract. Okay, a little fumble through acellular life. Sorry. Really not, I love that you guys have questions. It's not something I've ever been that into, which is why I probably haven't really focused on it. We'll skip organism of the week for right now. This class starts at 12:30. Okay. And go ahead and talk about prokaryotes. So again, prokaryotes are something that we know about in this class. How many of you have taken microbiology? The lady's talking in my ear. Okay, we're reconnected now. Temporary technology that I don't think I did anything to cause that. So prokaryotes are as we mentioned when we started talking about the diversity of life, this thing that I don't think we really have any true grasp how many prokaryotes are out there, right? We can estimate, but we're discovering all the time that prokaryotic diversity is probably a lot more complex than we originally even imagined. We don't really know how to define a species because we have both vertical and horizontal gene transfer happening. And so what is a species when that's happening, right? When you traditionally think that you can't give genetic information to unrelated individuals, and these guys can. So they're a pretty fascinating group of organisms, and a place where I think that mean, we are currently doing a ton of research into prokaryotes, and I think that will just continue, and we'll continue to sort of, as our genetic skills become more savvy, I think we'll begin to understand a little bit better what's happening and how many prokaryotes are out there. The color in this, I don't know how many of you have ever been to Yellowstone. The color in this thermal hot spring is due to archaea, which are prokaryotes living in this extreme environment. But this is a very extreme little puddle of water. It's extremely hot and has probably a pH that would be unreasonable to much life, may have really high concentrations of metals or chemicals that are toxic to us. And in these extreme environments, archaea particularly tend to thrive in extreme environments. But prokaryotes in general can live in environments exactly like the environment we live in, and then can also live in environments that are just really, really extreme for most of life. Okay, so your chapter has a lot of sections. We're going to cherry pick a couple of highlights from each of these sections. Okay, so I'm not going to cover any of these sections fully, and I'm just going to call this content chapter 22, but I'm just going to pick the highlights from each of those sections. The highlights according to me. Be honest about that. So we talked just now about viruses, right? Jerry did that. We're going to save this for later. Now we're talking about prokaryotes, which include these three guys. So even just in this really generic diagram, pretty big difference in size between individuals. The photosynthetic prokaryotes are much larger than the non-photosynthetic prokaryotes, but there's also shape diversity, right? And so some prokaryotes are round, some are oblong, some are corkscrew shaped, and that also changes how their colonies are formed. Okay, so in our tree of life, we have three main domains, right? Bacteria, archaea, and eukarya. The bacteria and archaea are all part of our prokaryotes. Okay, so big chunk of what we understand to be life is prokaryotic. The cells are simple, the cells are relatively small, and they don't have internal compartmentalization, right? There's no membrane around the genetic material. There are no organelles or separation of tasks or jobs within the cell in either of these domains. We're going to kind of talk about them collectively as prokaryotes, and then we'll talk a little bit about the differences between the two. In general, unless I specify, I'm kind of talking about all of eukarya and all of archaea collectively as prokaryotes. Okay. So as we sort of become a culture of humans who think about our own microbiome a lot more, I just love to remind us that if you actually look at the human body, we now estimate that the prokaryotic cells in and on the human body outnumber the human cells 10 to 1. So you're a whole bunch of water, a whole bunch of prokaryotes, and some human cells sprinkled in there for flavor, right? They're really, we are a host to so many different things. And most of those things are an integral, important part of our functioning, as opposed to a pathogen or something that's harming us. Right? Okay. So a couple of the highlights I want to hit are just about features of prokaryotes that are maybe different than or analogous to eukaryotes. And so the first is prokaryote communication. When we think about cell-cell communication in eukaryotes and in multicellular organisms, it's pretty similar. We use some sort of chemical signal, and that signal is going to send a message from cell to cell and potentially organize the cells to work collectively together, right, to do a particular task. And prokaryotes are able to do the same thing. So even though they're single-celled organisms and they aren't living as part of a multicellular body, they can actually communicate with each other and they can come together. And many of our prokaryotes spend most, if not all of their lives living in a colonial fashion. And so although they're not a multicellular body, they're living in really close association with other cells. They're communicating with those other cells. And in some cases, the collection, the colony, is behaving in a particular fashion together. So prokaryotic cells synthesize and secrete signaling molecules. And usually what happens is as they molecules, the concentration of signal increases in the solution that the organisms are living in. And so then other cells take up those excess signals. And so once the concentration of signal molecules gets high enough, the neighboring cells all start taking in those signals, and then the signals cause group behavior. And that group behavior is referred to as quorum sensing in prokaryotes. And basically it means that the organisms that are all living in that space together, or the colony, will behave collectively. And that's due to using signaling communication molecules. And so one of the collective behaviors that you can see is the formation of a biofilm. Does anybody know what a biofilm is? Huh? A movie about life. I love it. I always try to get you guys to use your root words. If we were using our root words, that would be a perfectly good definition. This is another definition of film. Instead of being a movie, is like a covering to a surface. Okay, so a biofilm is a prokaryotic community, so individual cells that secrete an extracellular matrix, and it forms sort of a sticky slime. It's a polysaccharide protein mix. It's a little bit gooey in some cases. It can actually be kind of hard in some cases, but it forms a film, and that film is sort of the body of the community, and then the cells are kind of embedded in their film, and the film can actually protect them from things on the outside that would harm them. So biofilms can protect the individual cells from things like antibiotics or from disinfectants or from cleaners. And so a place that we might be familiar with biofilms is on our teeth, right? You're supposed to go to the dentist, what, like every 6 months or something? Every 6 years, please. And they, I hate the dentist. And they scrape and scrape and scrape, right? When they're scraping, they're disrupting the biofilm. Okay, there's actually a biofilm that forms on the surface of your teeth, and the scraping is to help disrupt the film so that you can actually get in and remove that colony. Okay, all the other examples are even more disgusting to your teeth. I'll stop there. Okay, but biofilms are protective for the individuals that are part of them. They can provide resistance to that rather than a genetic resistance to antibiotics. They can essentially prevent the antibiotics from penetrating the cells because of the protective film. That makes sense.
So, like in the case of the teeth, is the film like itself black, or?
Yeah.
So is that like the dirty crap, or is plaque covering the dirty crap?
Well, what we call plaque is the biofilm. It's the protein and the carbohydrate, and then the cells that are protected by the matrix. Well, and you remove the bacteria with the plaque, yeah. So as you remove the plaque, you're removing the biofilm and the community that lives embedded within it. Okay. Another type of biofilm is sometimes referred to as a microbial mat. And microbial mats kind of, I think that the only reason we have a different definition for it is that they can be really big. They can actually be really, really thick. Like usually when we think about a biofilm, we're thinking about a thin layer like plaque on your teeth, or that little bit of sliminess in the dog water bowl or whatever. It's sort of a thin layer. But microbial mats can actually get really, really large. And so a microbial mat actually has multiple sheets of prokaryotes, almost like tissues, but not really, because they're communities of independent living organisms. But you sort of have these layers of thick biofilm that cover surfaces. So this picture here is from your textbook, and this is actually of a really big microbial mat that is in a deep sea vent. And the mat itself actually has openings in it. And so this didn't blow up very well. But there are these little openings that are actually allowing air and water and stuff to move through and over the mat. Like biofilm to biofilm? I don't know. I would bet that people have studied that, but I don't know any, like I don't have anything to offer there. But I do know that like these microbial mats are not necessarily always a single species. So you can have multiple species coming together and forming this aggregation. And so then the different species are benefiting from the sort of collective behavior. Okay. Again, the very broad question of what is a species in prokaryotes, because they do a lot of things that species of other organisms don't do in terms of how they share materials and work together. So lichens are separate, and we'll talk about them later. They are not they are neither prokaryote nor fungus. They're a symbiotic association of either a cyanobacteria or an algae and a fungus. So lichen actually isn't a single being. It's a symbiotic relationship that forms the lichen. We'll talk about them when we talk about fungus. Okay. Also, some of our prokaryotes are mobile. Not all of them. Some of them have to sort of go with the flow, right? They don't necessarily have the ability to move. All of our prokaryotes are going to be at least found in environments that have some moisture or dampness to them. They're not found in extremely dry areas. They always are going to have a little bit of moisture to that environment. And so some of them have the ability to actually swim through fluid. I put this picture up here only because see flagella on prokaryotes, it's very easy to just assume that it's just like eukaryotic flagella. And functionally, it works the same way, right? We have a whip-like thread that allows the cell to swim through an aqueous environment or through fluid, right? But structurally, it's not the same structure. So the flagella of prokaryotes has a different underlying structure. It's not made of repeating units of microtubules like that of eukaryotes is. So it's different. It's not like one of those homologies that comes up through the tree of life. It's a different structure to do the same purpose of swimming. Okay. So some individuals are able to be mobile, others drift or go with the flow, right? They just move around wherever the environment takes them. And then those things that form biofilms or that attach actually adhere to surfaces. And so in that way, it can become anchored. And so if you form a biofilm, even though the water is flowing around you or the medium is moving, you're staying in place. So you might attach close to a food source, or you might attach in a place that's safe and protected, or a place to reproduce, right? And so you would have a way to anchor yourself and not move, okay, which can also be an advantage. Okay, and then bacteria also have the ability to form something called an endospore. We're not going to talk about these analogous structures that much as we go up through the eukaryotic life, but this is not an uncommon strategy for surviving tough times. Okay, so lots of organisms have some sort of way to sort of hunker down and persevere during environmental conditions that are not good for you as a species. And for prokaryotes that are rod-shaped, we'll talk about the shapes in just a minute, one of the things they can do is form an endospore. And so this picture over here is showing us a rod-shaped bacteria, kind of like our E. coli. And then the red part here is the endospore. And the endospore is basically like a really protected package that the cell is going to put its genetic material in, and the enzymes it needs just start growing and reproducing. And so it's going to put all of that in a protective package and thicken it and reinforce it. And then that endospore can survive pretty extreme conditions that are outside the survivability of the cell as a whole. So a cell that maybe normally lives at pretty typical environments for human being, if that cell forms an endospore, it could survive boiling for more than an hour. It could survive extreme UV exposure. It could survive, in some cases, 250 million years encased in rock. They've actually opened up some rock layers and had viable endospores be able to start reproducing and moving forward after having been encapsulated in rock for a really long period of time. So as soon as the conditions get good again, it gets cool enough or warm enough or moist enough or whatever those environmental needs are for that species, when they return, the spore opens up and begins metabolizing again. And so it can actually replicate, produce a new cell. You're off and running. Does that make sense? You guys still writing this? I know this is like the home of the wordy slides.
Something just discovered in the ocean. Someone just discovered something in the ocean about a burial ground.
I don't know, somebody's always discovering something about the ocean. I didn't read about that one. I think I saw something go by, like the title, but so I want to talk a little bit about how we classically thought about prokaryotes, and then now we are going to just sort of change that up a little. Again, prokaryotes are really, really small, right? You guys looked at cyanobacteria last week. You feel like you could see those anabaena and gliocaps and see all the details and really understand what was happening in those animals' bodies? No, right? Like, that's like, you were just like, that's a really big prokaryote. And you're just like, I see a green spot, right? You can't really visualize them. And so because they're small and really hard for us to visualize their detail meticulously, even with things like electron microscopy, we are, you know, maybe not able to understand their detail as well as we can of larger cells. And historically, it's based on their shape, if they move and how they move, what pigments they make. So like photosynthetic bacteria, what different types of photosynthetic pigments are they making and using? What are their nutrient requirements? Do they form colonies? If so, how do they form their colonies and interact with colony mates? And then how do they react to stains that we use to visualize them in the lab? Right? So if you hear the terms gram-negative and gram-positive, that has to do with how the individual cells react to a staining procedure we use to help visualize them in microscopy. Okay? And so all of these things have been used to categorize prokaryotes historically. And so the three most common shapes that we've sort of broken all prokaryotes into one of these shapes is to be spherical or cosi. So any of the Staphylococcus, the COCC OUS or COCCI refers to being a spherical bacteria. So when you hear that term, you're looking at a bacteria that is a round shape. The rod-shaped bacteria are sort of long and oval and shaped more like a rod and less like a berry, right? And then there's the corkscrew-shaped, and the bacteria that are corkscrew-shaped tend to be a lot harder for us to treat as a pathogen because they can actually bury themselves into tissue. So things like the bacteria that causes Lyme disease is a spherical or corkscrew-shaped bacteria, and it can, excuse me, actually embed itself down into joint tissue, and then like whatever we want to throw at it, it's kind of protected, just like you'd be protected if you were part of a biofilm, right? So those three shapes impact how they interact with each other, impact how they interact with potential host organisms, and it's one of the like classic things we've used to broadly categorize them. Now we really know that what we need is molecular biology to classify our prokaryotes because they can exchange genes horizontally, right? Okay, I'm going to briefly just mention a couple of things that are different between bacteria and archaea. If you just saw a picture of one, I don't know that you would go, oh yeah, of course, obviously that's an archaea, right? These two beasts look pretty similar. One's in black and white and one's in color, but other than that, they have similar shapes. They both have flagella for movement. The top individual is a bacterium. The bottom individual is an archaean. So there are some things that are similar and some things that are different. Cell type, they're both prokaryotes, right? But they do actually have, and their technology is kind of all over the place. It's not like it's consistent one way or the other. But they do have some differences in their cell wall. So bacteria have peptidic glycan in their cell wall, and archaea do not. Okay, so there's a structural component of the cell wall that is different. And then there is a difference in the cell membrane type. So bacteria have a lipid bilayer. Everybody has a lipid bilayer. Archaea could have a lipid bilayer or could have a monolayer. Okay, which also seems kind of weird when you think about like how does that work. And then they have differences in their plasma membrane lipids. So your book has a nice picture that I didn't add in here. Bacteria have fatty acids for their plasma membranes. Archaea also have fatty acids, but they have these extra groups that change the shape of the fatty acid tail and therefore change the structure of the archaea cell, lipid bilayer or monolayer. So you have mostly differences in the cell membrane and differences in the cell wall. The outer surface is what's the most diverse. Okay. Huh? Huh? It's also a modal. Good enough? Need like one last word. Okay. Prokaryotes collectively, whether we're talking about archaea or bacteria, all for metabolism. Okay, they can be anaerobic or aerobic. Right? Anaerobic means that they can metabolize without oxygen, and aerobic means they require oxygen. So if you're an aerobic organism, you require oxygen for metabolism. If you're anaerobic, then it depends what kind of anaerobe you are, whether you can metabolize without oxygen or whether oxygen is actually toxic or lethal to you. Okay, so those things that are obligate anaerobes are actually poisoned by oxygen. And so you're only going to find them in environments where oxygen is not available or not readily accessible. So you find them usually inside some sort of barrier that protects them from the atmosphere. So they are down in the muck, submerged in something. You can have them in certain parts of the body where they're actually in these interstitial layers kind of tucked in around organs where they can actually be relatively isolated from oxygen. Like can have anaerobic bacterial infection in your gallbladder is one example. Otherwise, a huge number of prokaryotes are what we call facultative anaerobes, which means they have their choice. When oxygen is low in availability, they go anaerobic. When oxygen is available, they use it. And so a bunch of species will have higher rates of reproduction and may do more swimming behavior if oxygen is available because they have more energy available to them. And then if that oxygen is taken away, they become more sedentary, they reproduce at a slower rate, but otherwise they're fine. And so depending on the species, they can change it up a little bit. And then when it comes to metabolism, prokaryotes also have all the different options. Okay, they might be autotrophs. And here we have prokaryotes that can be photosynthetic autotrophs or chemosynthetic autotrophs. And so they are able to produce their own energy, either by using chlorophyll or other pigments to harness light and turn that into sugars, or by using the ability to harvest energy from inorganic chemicals. And so the bacteria that actually live in the velvet tube worms that make the tube red are actually feeding on sulfur. And so they're actually taking the energy from the sulfur element and using it to power their own metabolism. Okay, so the chemosynthetic prokaryotes are pretty fascinating because they can do things that, again, we really don't see eukaryotes that can just take sulfur and use it as an energy source, right? Especially some of the things that live in those extreme environments are adapted to utilize those extreme environments through chemosynthetic metabolism or creative pigment for photosynthesis that we don't see in other organisms. Okay, I think, yeah. Let me just finish up this slide and we'll stop here and we'll just do the last little bit of prokaryotes whenever we see each other again, whether that's Thursday or not, okay? So prokaryotic reproduction or division is pretty similar conceptually to what we see when we think about eukaryotic mitosis. It's just simpler, okay? We actually talked about this in Bio 1. The cell undergoes what was historically referred to as binary fission. We now call it prokaryotic fission. It's an asexual process where the cell makes of itself. Okay? So when the prokaryote actually divides, it's going to begin by copying its circular chromosome. If it has plasmids, those plasmids can actually copy independently of the genomic chromosome. So the circular chromosome copies. The chromosome is actually attached at the origin of replication to the plasma membrane. So then the plasma membrane begins to grow. And as it grows, the chromosomes move with the plasma membrane to the poles of an elongating cell. And then it pinches in half and builds a new cell wall between the barriers. Okay. Prokaryotic division can be super fast. In E. coli, optimal conditions, once every 20 minutes. They can just keep doubling their population. So it allows for rapid population growth. It also allows for rapid evolution, right? If a mutation arises, it can be propagated through the descendants really, really quickly. Okay. So we'll finish up this chapter and talk about that horizontal gene transfer piece. How do we get genetic material between individuals, not just making clones of ourselves? And then in our next lecture, we will start to talk about our eukaryotic life. Okay? All right, so take 15 minutes, come back to class, settle in. We have a lot of activity going on in lab today, and so